let's get started on the nitrogen cycle. So what I'm going to cover, what my learning intentions are this evening, is that we need to ask each other these questions. Can you give the role of bacteria in the nitrogen cycle? Do you know the significance of nitrates in producing proteins and nucleic acids? You might want to think about the element nitrogen there. Can you give the importance of human activities such as ploughing and drainage in producing the aerobic conditions needed for nitrification, an important process in the cycle? And then finally, do you know the economic importance of the nitrogen cycle in relation to food production with a growing world population and fertilizer application? We'll be looking at the different sorts of fertilizers there. So what's nitrogen used for in organisms? Well, let's have a little look at this photo to start with. I don't think it's going to take you too much to realise there that what we've got here are two plants that actually have grown quite differently. If you have a look at the plant on the left-hand side, it's grown significantly more than that on the right-hand side. And this was a well-controlled experiment where we used the same species of plant, they were grown for the same length of time. Everything about this experiment was controlled, apart from the fact that the plant on the right was grown in a water solution that didn't have nitrogen in it. So I think you can probably see the effect that that has on growth. Now, having said that, then we know that nitrogen is important for growth, but what is it used for as well as to grow? Well, we know from our work at GCSE that nitrogen is used to make amino acids, and from there they're the building blocks of proteins. And we learned at GCSE that nitrogen was for growth and replacement. So we know there's something about that. Also, from AS level as well, we know that we find nitrogen in nitrogenous bases, that's DNA, RNA, not forgetting that ATP is a nucleic acid as well. So you find nitrogen there. And obviously you'll connect that with active transport. And then also recently in Unit 3, you'll have covered photosynthesis, and you'll know that nitrogen is at the very heart of the chlorophyll molecule. So just a couple of images there for you to sort of recap. The top one there, you can see the nitrogenous bases, adenine, thymine, guanine, cytosine. I'm sticking to DNA there, obviously not talking about uracil for RNA, and you'll see the general structure of an amino acid there, okay? And then the bottom diagram, you can see, even though we've got magnesium 2+ ions at the centre of this chlorophyll molecule's porphyrin ring, you can see also that there are nitrogen atoms there. So, we know nitrogen is pretty important to make all of these substances. Okay, so how do plants actually absorb nitrogen then? Well, they absorb nitrogen in one of two forms, that's ammonium ions and nitrate ions. There's a slight problem though, in that plants don't have an enzyme to break the nitrogen-nitrogen triple bond. So they cannot take nitrogen. You think there's a plentiful supply of nitrogen in our atmosphere, but they haven't got the enzyme to break that bond, so they have to take it in these two forms. So what happens is that they have to have it in a fixed form. Okay, and the fixed form means when nitrogen is combined with, for instance, in this case, hydrogen and oxygen atoms, okay, and it turns it into then what we'd call an organic compound. So yeah, it's a very unreactive gas, is nitrogen, but plants need a source of it anyway. They take up the form of nitrogen they can use either by active transport or facilitated diffusion. And if you think about it, if you think about your work with maybe nitrate, you know that it depends on the concentration of nitrogen in the soil as to whether they will take it up with the concentration gradient by facilitated diffusion using the proteins in the cell membranes, or against the concentration gradient by active transport. And also because, I've just mentioned that there are proteins in the cell membranes, I've mentioned active transport and the connections there with ATP, you can see how important it is for the plants to actually take up a source of nitrogen, because obviously nitrogen is used in ATP and it's used in membrane transport as well to make the proteins in the membranes. So as well as all of that, we know that protein synthesis is going to be affected by the absorption of nitrogen and obviously its ability to make chlorophyll as well. So moving on, there are, I would say, 4 distinct parts to the nitrogen cycle. There are a lot of exam questions on these parts. So we'll be talking about nitrogen fixing, nitrogen fixation. We'll talk about decay, decomposition, ammonification, putrefaction. You know, why use one word in biology when you can use a lot more? That's what we like, isn't it, as biologists? So we'll talk about those processes together. Then we'll look at nitrification. And finally, we'll look at denitrification. So here we go. Let's look at the first one. So nitrogen fixation or nitrogen fixing. This is where we combine, or a nitrogen will be combined with an organic molecule. So this is done in a number of ways. So there are what we call free living bacteria in soil. Now those are, the example that we most commonly use are Azotobacter species. Okay, I've written it in italics there for you, as you'll be familiar with. And they live in the soil. They don't live in an organism, they live purely in the soil. And what they do is they convert all of the atmospheric nitrogen, which is in the soil air spaces, into ammonium ions. Now, ammonium ions, if your chemistry isn't sort of as good as you'd like it to be, and you can't remember all of these different ion symbols, then ammonium ions, learn to recognise that NH4 plus is an ammonium ion. But if in the exam you're getting yourself a little bit sort of, tongue-tied, a little bit knotted up, then please make certain the ammonium ions word is in there. So free living bacteria in the soil. And then we come to a bit of French, legumes, leguminous plants. They are peas. I need to say green beans there as well, okay, not, baked beans. So leguminous plants are things like peas, green beans and clover. Now, they actually contain another bacterium called rhizobium, and this is a nitrogen fixing bacterium. And it actually converts, just like Azotobacter, nitrogen to ammonium ions, and then from there into proteins that the plants can use. We mustn't forget the chemical process, the Haber process, where there's a massive commercial process there where nitrogen is converted to ammonium ions and nitrate ions as well in fertilizers. Okay, so what happens is that actually ammonium nitrate gets produced, as a fertilizer. And as I say, with an increasing population, feeding the world's population is certainly a major consideration and big business. And then the final process, lightning. And we're not really going to talk about Haber process or lightning here today. We're going to focus on the other two, but I wanted to give you the complete picture in any case. So that's nitrogen fixing then. So, how do they do it? Well, the bacteria have this enzyme called a nitrogenous enzyme. It's a nitrogen reductase actually, and what happens is that if they have this bacterium, if they have this enzyme, then they're able to convert the nitrogen gas into ammonia or ammonium ions, preferably ammonium ions because ammonia is toxic. Now, these are either free living in the soil, the Azotobacter, or they're in the root nodules of the leguminous plants, rhizobium. So those bacteria are very useful because they can fix that gas. The other thing is just to tell you a little something about how the bacteria actually got into the root nodules or get in there. You may be able to see in this image here, I mean, there's sort of a pink part of the image here and a green one over here. And this shows how it is that the bacteria can infect the roots. Now, I'll just trace around the bottom. This is a root hair. You might be able to see around the green parts, the actual root hair. And as I've tracked down, you might be able to see the sort of rectangular shape of the root hair cell, the rest of it there. And what happens is the bacteria then, as I say, enter the plant through those threads in the root hair cells. So that's how they get in. So that's rhizobium. Now, there is another word I'm going to introduce here, which is mutualism, because what happens is you might think, well, why is the plant able to survive if it's being infected by bacteria? Well, they're not pathogenic, as this turns out, they are mutualistic. So we need to think, how is it that rhizobium benefits? How does the leguminous plant benefit? Surely there's got to be some sort of equal relationship here. Well, in fact, the rhizobium receives amino acids from the plant. So the plant is able to make its own amino acids, its proteins, its carbohydrates, and a whole host of other organic chemicals that this bacterium, this rhizobium, can benefit from. And the plant itself, and you can see in that image here, you can see this sort of, I'm just circling now with my pointer, the root nodules. The plant will be able to have the plant has a supply of ammonium ions, okay, from the rhizobium. So it's a sort of a mutually beneficial relationship there, okay. So that is why the rhizobium is able to survive in root nodules, but also thrive. Now, the second process, as far as nitrogen cycling is concerned, is this one that I said, well, why use one name when you can use, a number? And this is decomposition. And what happens is that when bodies die or they produce waste, what happens is their body decay. And organisms like fungi, you can see I've just shown you some images there on the right hand side, some fungal hyphae. What happens is that they decompose living things. Now, organic nitrogen, what I mean by that is like a dead body of a plant or animal. And here we go, here's urea, so that will be a waste from the urine of animals. And what happens is that in both of these cases, saprotrophs, as I say, fungi, bacteria, will convert either parts of the dead body or the urine, the waste urea there, into ammonium ions. Okay, so that is basically decomposition. Now, obviously, we don't, we want to produce ammonium ions because this is the form in which the plants will take it up, not ammonia, because that's toxic. The third process then. So this is the one that involves multi-stages, and we call this an oxidation process, nitrification. And we say it's the oxidation of ammonium ions into nitrates. And we're going to talk about chemoautotrophs. Okay, because that's what the bacteria are living in the soil. So we'll start with the protein, which is available in living things, and then what happens is they can decay, as I've just said. Now we, as well as the word ammonification, converting into ammonium ions, we also use the word putrefaction there as well. So we've got a range of organisms here which will bring about the decay. So I've talked about the saprotrophs. What I didn't talk about was detritivores, because organisms like worms, what they do is that they take big bits of dead organisms, and actually what happens is as it passes through their digestive tract, they produce smaller bits, which then the bacteria with their enzymes are able to digest a little further. So first stage here, what happens is that through this decay and decomposition, we've now produced ammonium ions. Next stage. Now this is where we start with a bacterium called Nitrus amonas. And I'm going to draw your attention to how to actually remember Nitrus amonas comes before Nitrobacter, which is the next one I'm going to talk about. And this is the nitrification process. So what happens is the ammonium compounds are oxidized. What happens is they take on oxygen, the nitrogen is removed, and what happens is that nitrates are formed, NO2 1-. And then along comes another bacterium, and it actually converts the nitrites, the NO2- into NO3, the nitrates. Now, these bacteria, Nitrus amonas and Nitrobacter, actually are aerobes, so they need a supply of oxygen in the soil. And actually what I was mentioning earlier on was that the names of the bacteria, so I always think Nitrus amonas with the A always comes before Nitrobacter with the B, and that's the way I remember it. And of course what happens is you take on more and more oxygen as you go through, so it's ammonium compounds to nitrites to nitrates. So I'm hoping that's sort of a usual way for you to remember it. And then the fourth phase in the nitrogen cycle that I'm going to talk about is denitrification. So this is a real destructive method, this is, and you can see I've shown you a little image there of fields, and they're absolutely waterlogged. I was only looking at a plant pot of mine earlier on and thinking exactly the same thing, because what's happened is the soil has been compacted, there's lots of water about, and the water can't drain anywhere. So if a field isn't regularly ploughed, if the water is allowed to settle on the top like this because the soil underneath is quite hard and it's not being turned, then denitrification occurs. And so all that wonderful nitrate that these bacteria, the Nitrosomonas and Nitrobacter, have been making is now converted back into nitrogen gas, because these waterlogged soils actually create an anaerobic condition there for these denitrifying bacteria to work. Okay, so it is worth also mentioning at this point about something called leghemoglobin. I'll show you a little image now. Let me just there we go. That's the image I want to show you, because you may have to make some links between leghemoglobin. You can see it's a lovely, lovely red colour. So it's no surprise that there's a hemoglobin type derivative there called leghemoglobin, and that associates with oxygen really rather well in the root tissue. Now, some exam questions ask about it, but what you have to remember is that actually it's not aerobic, because what the leghemoglobin does is it associates with oxygen, and that provides better conditions, ideal conditions for the rhizobium to carry on its work there, okay, and producing ammonium ions, nitrate ions for the plant. So don't be misled by the fact that you may see something that looks like hemoglobin and immediately think, is the question about aerobic in some way because of the oxygen? Well, just be very, very careful, and also make links for yourself there between the fact that we've oxygen and ATP are both needed for absorption in the roots. And obviously the oxygen conditions are going to be low there as well. But of course, ATP cannot be made in high enough quantities if we don't have aerobic respiration. So just bear that in mind when you see exam questions, which there are on leghemoglobin around. The next thing I want to go on to really is how it is as humans we actually affect the nitrogen cycles, because we do as humans, we have a big impact on the nitrogen cycle. I've mentioned about ploughing. Ploughing is really, really important, because what will happen is the soil is turned around, so it'll aerate the soil, and that will actually cause the mixing of oxygen from the air into all of those air spaces that I talked about earlier on. And of course, that's so important for those aerobic bacteria to carry out nitrification, the Nitrosomonas and the Nitrobacter, okay? And then the other process that actually humans influence is this idea of draining, because what happens is that in farming you need to make certain your soil is adequately drained, because remember I said if actually it's not drained, if you get this waterlogged soil, then you don't have aerobic conditions. So what happens in a lot of farms is that farmers will actually dig drainage ditches on the edges of their fields, and any excess water, if we've had a massive downpour of rain, what happens is the water will then drain off the field into the drainage ditches. And of course, farmers have got to keep those well maintained, in order to ensure that the water carries on running off the fields in this way. So both of those activities are really, really important to promote the nitrogen cycle, and particularly their nitrification, because it's an aerobic process. But there are other ways in which humans affect the nitrogen cycle, and this is, I mentioned this earlier on, artificial fertilizers. Now, artificial fertilizers are things like ammonium nitrate, but also there are natural fertilizers like manure and slurry. And all of these things will add nitrates to the soil. Okay, so you can see here that there's in an industrial farming situation, you'll see that the farmer is spreading quantities of artificial fertilizer, ammonium nitrate, into the soil, and this is soluble, and that creates its own issues, because if actually the farmer doesn't get the spreading of the fertilizer just right, what will happen is you could have a downpour of rain and all of that lovely ammonium nitrate is washed into those drainage ditches I've talked to you about. And therein lies a connection with another topic in biology, because if you're not careful, and if those drainage ditches aren't maintained, and if the farmer spreads the fertilizer when it's raining, you can set off a eutrophication, which has its own issues, but we haven't really got time to chat about that today. So the other thing about when you're spreading fertilizer as well is you shouldn't spread it too close to the edges of the field, because then any runoff, any water will cause it to run off into the watercourses, the drainage ditches and local ponds, etc. Okay, we call that runoff, or we sometimes call it leaching. But of course, this is all in conflict, isn't it? We need to manage this very, very carefully, because we have an ever increasing population on the planet, and actually that means we've got an ever increasing demand for food. And as much as we might like to think that we can keep up with the demand to feed people using natural fertilizers like manure and slurry, we can't always. Okay, so that is how our human activities will, affect the nitrogen cycle. Now I've put this slide in. Oh, I'm just going to go back. I'll just say something briefly about the previous slide, and that is that I might come back onto this later, which is in developing countries that they may plant peas, beans and clover in order to naturally fertilize the soil as well, because obviously ammonium nitrate costs money. So I might come back to that a little bit later if I've got time, but the reason I've put this slide on it, you will have seen lots and lots of different images of the nitrogen cycle, I'm sure, and I haven't put this in because this is the perfect one, but I've put this one in just to say to you, do you know, once you've got your 4 processes sorted and you've got each of them sorted in your mind, then just take a piece of paper, take an A4 or an A3 piece of paper and start, I suggest maybe start with how nitrogen is absorbed by plants. Start with that and then show how it is, from what we've just been talking about, that nitrogen can be cycled through the plant into animals who will consume. Then what happens to that nitrogen when either the animals produces waste, nitrogenous waste like urea, or when the plants and animals die, what happens then? What happens to all of the ammonification, the putrefaction and so on? And then from there, once you've produced ammonium ions, what happens in nitrification to actually then channel that nitrogen back to the plants? Not forgetting the role of our nitrogen fixers, our really important rhizobiums in the root nodules, in the leguminous plants, and the nitrogen fixers. Okay, so all of these things together, will combine to improve your understanding if you do it for yourself. Okay, so that's just sort of a little suggestion for you. Okay, let's get on with some questions then. So what we have here in front of us is a photograph which shows root nodules on a plant, and you can see those. It's a black and white image, but you can see the root nodules there. And it actually starts by saying, name the group of plants which have large numbers of root nodules on their roots. And I said earlier on, well, I'll probably be pausing. All right, so just literally a few seconds. Can you think of the name of the plants? Okay, so well done if you've thought, well, it was peas and it was green beans, or it could have been clover, or I could have actually said legumes. So well done for that. And then the next part, explain the advantage to these plants of having root nodules. So we've discussed parts of that. So just in your mind now, talk to yourself about what it is that actually means that these plants have an advantage, all right, because they've got the, they've got the bacteria in the root nodules. Just a couple of seconds for you. Okay, so let's see then. Okay, so those nodules contain the nitrogen fixing bacterium called rhizobium. Okay, so that's pretty important. And the thing about rhizobium, remember, let's make our connections again now, because we've got 3 marks here. So we want to talk about nitrogen fixation. We want to talk about an aerobic bacterium like rhizobium, and the fact that it's supplying the plants with ammonium ions, and we'll come back to this later on. So the plant then can be grown in nutrient poor soil, and it's just what I've said. If actually in a developing country there's not the money to buy artificial fertilizer, then in your field of crops, in between your crops, you can actually grow some peas or bean plants, because then there will be a sort of a glut of ammonium ions for the plant. And then the next question here, which is nitrous amonis and nitrobacter. There we go. A followed by B are 2 groups of bacteria which carry out nitrification. Explain this process and why it is important to soil fertility. So again, 3 marks. Can you think about 3 things that you might want to say there? Okay, so have a little pause. Okay, and let's have a look then. So basically we know about nitrous amonis, but remember, because it's A, it converts ammonium ions to nitrites, and then nitrobacter will convert nitrites to nitrates. Okay, and then the plant obviously then can absorb the nitrates to be used in protein synthesis for chlorophyll, DNA, RNA, ATP, for active transport, etc., etc. Okay, so that's the first question. Now I'm hoping that was fine for you, bearing in mind everything we've just done. So let's look at another. Okay then, so we're on a roll now then. So nitrogen is required for healthy plant growth. Okay, we know that. We saw that image right at the start. Identify the following. 2 forms in which nitrogen can be absorbed by plant roots. So I'm just going to pause. Okay. Ammonium ions and nitrate ions. And this is sort of a classic example of what I was saying about, well, if you're more confident about writing ammonium and nitrate ions than writing the ions themselves, that's fine. But if you're going to write the ions out, make certain you write them correctly. Okay, it's not ammonia, it's ammonium ions, remember. And the next question says, identify the stage in the nitrogen cycle that will be slowed down by ploughing and drainage of soils. So the stage in the cycle that would be slowed down by ploughing and drainage. Now think about that. Ploughing and drainage, we want that to happen, don't we? So what stage is going to be slowed? And that is denitrification. Brilliant, I hope you got that correct. Because, ploughing and drainage is going to slow it down, so that's why we encourage it. And then one last thing, free living bacteria involved in the fixation of atmospheric nitrogen. Here we go, Azotobacter. And the emphasis in that question is free living. Yeah, so that rules out our rhizobium straight away, because that's not free living, it's living in those lovely root nodules. Right, moving on to another then. So nitrogenases, they're enzymes used by some organisms to fix atmospheric gas, that's nitrogen N2, into a form of nitrogen available to the plants. The NIF gene is the gene coding for the synthesis of nitrogenases. Again, enzymes, they get everywhere. So they're found in nitrogen fixing bacteria is this NIF gene. Rhizobium is a mutualistic nitrogen fixing bacteria. We're fine with that, we know that. It forms a relationship with leguminous species, legume species, and in some species of rhizobium, the NIF genes are located on plasmids. Now, this question isn't about plasmids. You learnt about plasmids last year. If you remember, plasmids are just a circular ring of DNA in that procell. Okay, so it's not the DNA, the naked DNA, which makes it a procell. It's the circular bit of double-stranded DNA in a circle. Okay, so, but that's not what the question's about. The question is, name the form of nitrogen produced by rhizobium that is available to plants. So again, a quick pause. Okay, and did you get ammonium ions? Okay, well done if you did. And then another one, name another genus of nitrogen fixing bacteria. All right, so quick pause. It's time to get the next genus of bacteria out. Azotobacter. Okay, brilliant if you've got that. And then precisely where rhizobium would be found in the legume. So you can't just say in the roots, that wouldn't be a good enough answer. So what are we going to say here? We're going to say, that's right, in the root nodules. Okay, so you need to mention the roots exactly. And then suggest how the relationship between rhizobium and a legume species is beneficial to both. So obviously this is the ideas that we talk about, you know, about it being a mutualistic relationship where they're both benefiting from it. So let's now just recap what I'd said earlier on. And, you know, that is, for instance, that the rhizobium produces the ammonium ions in the plant's nodules, which the plant can then use to produce its proteins. And of course, the plant is a very, very rich supply then of amino acids. It's got carbohydrates, it's got a whole host of other things that these rhizobium bacteria living in the root nodules can actually survive on. So this is how it is that the relationship is mutual. It's a mutualistic relationship. And I'll just say one last thing, just putting in again about the leghemoglobin. All right, the plant has this in its nodules. It associates with oxygen, so rhizobium can work in those anaerobic conditions that they positively thrive in. Okay, so don't forget that again. And another few questions. So if we have a little look at this one then, this talks about bacteria living in the soil, and actually this one talks about, sorry, I can't quite read my my question at the top there, and it says a denitrifying bioreactor is a trench in the ground packed with wood chips. It becomes colonized by soil bacteria that convert nitrates in drainage water to nitrogen gas. Now we know that's our destructive process. And then it says wood chip bioreactors may be a cost-effective farm scale practice for nitrate removal from agricultural drainage. When placed strategically, so in the right place, these bioreactors can reduce the nitrate levels. Okay, so it can reduce the nitrate levels flowing into streams and help most meet environmental demand, sorry, for the reduction of total nitrogen content in streams and river. So, you know, there's quite a lot of information there, isn't there? So actually, you know, PACT is suggesting to me that the conditions may be anaerobic, and, you know, this question is about denitrification. Well, it's talking about that as well. And obviously, we've got another link with eutrophication. And of course, this is what you learn to expect on biology papers, isn't it? You're not going to get a question that's just only about the nitrogen cycle, that's solely about that. It's actually going to be drawing you in with your big picture and sort of encouraging you to pull bits of knowledge from other places. So, okay, let's look at the first question. What is the correct term for the process when nitrate is washed from the land in the drainage water? Okay, so it's washed, it leaves the fields. Remember I said, if it's been raining, it could actually be washed out into those drainage ditches that I talked about. So have a little think, and it is, that's right, it's a runoff or it's leaching. Use either one of those. Next question, name one source of the nitrate on the farmland. Okay, so what is being put on the farmland? Not too difficult, this one. The answer is, that's right, it's fertilizers, or you could say manure, or you could say slurry. And actually I will just a little sort of shout out for slurry here, because I think we all know what manure is, but slurry is the kind of sort of waste solids in a lot of water. All right, so if you live in a farming area, you very often will see the farmers shipping huge tankers of slurry from you know, field to field. Okay, and then this next question, okay, we're all teed up for this now because it talks about our various soil bacteria. Ah, right, now we know our soil bacteria could be rhizobium, they could be azotobacter, they could be nitrosomonas, they could be nitrobacter. So we've got a few bacteria now that we can draw upon. But this says it can change ammonia or ammonium into nitrite or nitrite into nitrate as part of the nitrogen cycle, and bacteria of the genus X are the primary converters of ammonium into nitrite, and bacteria of the genus Y oxidize nitrate to form nitrate. So, you know, I don't think it's going to be too much for you to realise that, again, we're going A followed by B, and then why is it important for the plants that ammonia is converted to nitrate? Well, here we go. You know, we don't want toxic ammonia. Nitrate is not ammonia. And we know that nitrate is the main form that the plants will absorb. And we've talked about active transport and facilitated diffusion. So it's used to make amino acids and proteins. And then just one final question here. The drainage is really important for growth. Explain why farmers want to drain their land. Well, they don't want denitrification, do they? So they want to maintain the fertility to encourage the denitrification. There's a couple more questions here, but actually what I suppose I'd say about these is that have a little look at your own time now to just go through see if you can think about what the best words are to go there, and then the next slide will reveal the answers, and they're all in order there for you, so you'll be able to see those for yourself. I just thought I would finish the questions for today by talking about how you approach QER questions, because I do those in a certain way. And actually what I would say is I've just made up a QER question here for you. The nitrogen cycle comprises many different stages which are linked together. OK, we know that now. Nitrogen is a very important element in living organisms, yes, and humans affect the cycle in various ways, tick, tick, tick. Use your own knowledge to explain these processes, right, and the significance of nitrogen to life. So, as I do with these things, I find that you've got to work out the three parts of the question. Now, I've given you a bit of a heads up there, haven't I, that actually you've got your 4 stages. You need to say what nitrogen is for, and we know how humans affect nitrogen cycling. So, you know, for top marks in the QER, you know, you've got to have your 3 parts. And the other thing I do is I actually say to my students, right, get all of the keywords down now on this topic. Think of all your keywords, get them together in a stack. Then it's up to you. That's the first thing, but then it's up to you. Do you actually then want to put all of those keywords in a sensible sentence, or do you want to think, right, what sort of things could I say about those? And let me just sort of get sentences in order from them. So it's up to you which order you do it in, but remember that you will be in the top band of marks if you've got an articulated answer. So it's clear what you're talking about. You've got sequential reasoning. That means 1 point follows on from another, which is why number 2 and number 3, the orders are pretty important, but do what works for you. Fully address the question. So that means actually make certain you've got all 3 parts and make certain there's nothing missing there. All right, so follow those sort of little tips. Now, what I just did, I'm just wondering now, I'm just going to finish by thinking, right, are you surprised now by any of these words that I'm actually gradually going to be putting up here? Because these are the key words that I would be thinking about as far as the nitrogen cycle is concerned. OK, as they go up, is there anything here that is a big surprise to you? And actually, I'm hoping at this moment in time, you're saying, no, nothing there is a big surprise. You know, nothing at all. I can answer this. And you certainly can. If you have got your keywords and you understand what those keywords are all about, you will be able to answer any question on any biology test. I promise you. OK, so I'm just going to finish here by just saying that there's a whole host of resources out there. I'm sure you have lots of resources from your own teachers, and I'm hoping that actually what I've done here has actually recapped the nitrogen cycle for you, and good luck with revising this topic. I really hope that it's helped.